All right, for our RNA notes, so our types of RNA. We're going to start off with what RNA stands for before we glue anything in. RNA is for ribonucleic acid, RNA. When we talk about the structure of RNA, it is a single strand. DNA had a double strand. RNA is its structure, it's single strand because it's a single strand, it's small and can leave the nucleus. The nucleus has little tiny holes in its nuclear membrane. DNA, since it's two strands, DNA is too big, it can't fit out the doorway. RNA is a single strand, it's small enough to fit out the little doors. Primary function of RNA. Our function, it's going to carry information from the DNA, which is stuck inside the nucleus, to the ribosomes to make proteins. And that's the name of our unit that we're starting, is protein synthesis. Our first foldable we're going to glue in is the one that says RNA versus DNA. Wait, and there's extra there. I didn't see that. Everybody good with the information up top? It's about to go away. So here we want to compare RNA versus DNA. We're going to start on the DNA side. We've been talking about DNA for the last 2 weeks. I want to go through a little review first. refresh your memories. All right, first thing about DNA that we're going to put down. DNA is double-stranded. There were 2 backbones. Second important thing we know, or that we need to know, is the type of sugar. DNA was deoxyribonucleic acid. It has a deoxyribose sugar. And the third thing, the nitrogen bases. We had 4 nitrogen bases. We had A or adenine. What does adenine pair with? Thymine. And we have cytosine. What does cytosine pair with? Guanine. Now over on the RNA side. So first thing, single-stranded. Second, we have a ribose sugar instead of deoxyribose, so the backbone's a little different. And then our nitrogen bases. We have adenine again. We still have cytosine, we still have guanine. Thymine goes away in RNA. We have a new base. Instead of thymine, we now have uracil. What letter do you think we're going to refer to uracil by?
U.
U. What do you think uracil is going to pair with? A, because we got rid of adenine, or we got rid of thymine. So it's going to pair with A. Next flap we'll need to glue in is the one that says how information is copied onto mRNA. You might have space at the bottom. If not, go to a new page. I'm going to be going to a new page. You can go ahead and glue in both or your next 2 foldables on that next page. Next, we want to talk about how information is copied and our 3 types of RNA. And it's going to make more sense. I should want to start with this one. I glued them in wrong, but well, I told you how to glue them in. And rats. Just thinking about the order that they were when I cut them out of the sheet of paper. But we're going to start down here. Our 3 types of RNA. When our 3 types, we have mRNA, tRNA, and rRNA. When you write these, the first letter is always lowercase. The next 3 RNA will always be uppercase. We can tell the difference. Really important down here with this one when we have rRNA. Really seems rather redundant, but it has a meaning. We're going to start with this first one, mRNA. And this is what it looks like. mRNA is long and stretched out. Here's our one single strand. That M stands for messenger RNA. Messenger RNA is the one that carries the information from the nucleus to the ribosome. Our next one we're going to have is tRNA. It's still a single strand. See this one empty squiggle? This is like the one backbone of the mRNA, only in tRNA it gets sort of folded up on itself. The T stands for transfer RNA. Transfer RNA is going to carry amino acids. Amino acids were the building blocks of proteins. It carries the amino acids from the cytoplasm to the ribosomes. And then our last one That R stands for ribosomal RNA. Its job is it makes, we take a couple of pieces of ribosomal RNA and they combine together to make the ribosomes. The picture that you have in the front, the little squiggles, are trying to show you the RNA backbones. So there's a big piece of RNA, a little piece of RNA that come together to make the ribosomes. Today we're going to talk a little bit more about mRNA. He is going to be really important for the next couple of days, and then we'll talk more about tRNA and rRNA. So back up to the next one. How is information copied onto mRNA? As in order for the information to leave the nucleus, it has to get copied onto mRNA. We have a process called transcription. It's very similar to replication. We will talk more about transcription tomorrow. Our RNA complementary base pairs are a little different though. So what does G pair with? C. And what did we say that A was going to pair with? U. So A goes with U. Instead of T.